Hello, Miriam, and thank you for the invitation to your wonderful course. And hello, everyone, wherever you may be. I'm delighted to talk to you about CAR T cells, CARs, and the therapies that are based on this novel form of instructing T cell responses. If you're interested, here are my disclosures, which really don't bear on this particular presentation. Here are the two first CAR molecules that the FDA approved as drugs back in 2017. They are what we named CARs some many years ago. They are receptors for antigen. Both of these are specific for CD19, a molecule that we had identified as a target of potential interest in B-cell malignancies. In their cytoplasmic domain, here you see another characteristics of these molecules, which is a composite signaling domain. These were the first two. The one on the left, the one we reported back in 2002, which combined the cytoplasmic chain of the CD3 zeta chain to initiate T cell activation, which we had found is not sufficient and needed to be amplified in some way, first using the cytoplasmic domain of C28 on the left. Dario Campana showed you could do that by also incorporating the cytoplasmic domain of 41BB. Before I go more into CARs, though, I would like to discuss a little bit the rationale for doing this and go back to the immune system, the origins of our immune system, which obviously evolved to protect us against a wide range of infectious agents, that threaten us as a species. And of course, we couldn't be around and reproduce if we succumbed from infections before the age of reproduction. And as you all know very well, the immune system is layered in many systems. And at the top, for some anyway, I don't know, at the top, is the adaptive immune response, which rests on the generation of highly specific receptors, either the B cell receptor, which can be secreted from B cells, or the T cell receptor, which is harbored by T lymphocytes. And yes, we know that this repertoire of T cells is generated in the thymus through genetic rearrangements, generating a repertoire of that some estimate to be of about 20 million specificities in an average human, at least by some estimates. This is a large repertoire, although it's not infinite. As McFarlane Burnett had predicted, this formation of these receptors needs to be pruned or purged of autoreactive T cells. And again, this repertoire is largely biased towards infectious agents. Cancer, however, is not an infectious agent. It is not made up of entirely foreign antigens. In fact, it takes its origin in our own cells, which acquire somatic mutations. And here I refer to this drawing that many of you would have come across long ago, that reports you on the y-axis the mutational burden that is found in different cancers. And to the right here, in which I circled in gray, are some cancers that have high mutational burdens. And that creates a large number of what we now call neoantigens. And the more neoantigens there are, the better chance that you might have an endogenous T cell response, or maybe an active immunization elicited response against one of those neoantigens. Now, some estimate that maybe 1% or so of all these somatic mutations in exons that you can identify might result in the formation of a neoantigen. And if really it is about 1%, well, you can tell that if there's fewer than 100 mutations, you may not harbor, this cancer may not harbor that many opportunities for the T cell receptor to engage one of those neoantigens. And so this is where CAR T cells come in. CAR T cells, as I just depicted for you, engage cell surface antigens through the SC of V, that is at their external domain. They are not HLA restricted, but they can also engage HLA negative tumors, therefore overcoming one of the escape mechanisms adopted by cancer cells to avoid T cell responses. And here, if you see my arrow here, I find them particularly relevant to cancers with low mutational burden, where again, just by probability, there's a lesser chance of encountering a neoantigen. My fourth bullet here is, as for any adoptive cell therapy, it may provide you rapidly, very rapidly, upon infusion, a large number of tumor reactive T cells, in contrast to the time it would take for a spontaneous or a response or a vaccine elicited response to kick in. So this brings us to these CARs that engage cell surface molecules in tumor cells or any cell. And these target molecules may be proteins, they might be glycoproteins, it could be a glycolipid, it could be a carbohydrate moiety on a protein. Now, this field is heavily predicated on genetic engineering. If you see my arrow here in the upper left, which started with retroviral vectors, and the approaches, of course, rely a lot on what we learned from tumor immunology, T cell biology per se, the microenvironment. But because these cells don't engage HLA peptide complexes, although they can be engineered to engage HLA peptide complexes, we're talking about a very different set of targets that must be, as I said before, on the cell surface. And CD19, this target that we identified as a possible candidate way back in 2003, we reported that you can target CD19 with CAR T cells, is a sort of a paradigm for this approach today. And there are many more cell surface molecules today being investigated as targets. The receptors, if I move here to at 3 o'clock on the right, as I already said, are not natural receptors. They're synthetic receptors, which need to be introduced genetically into these T cells. And to bring this to the clinic required the development of what you could call cell manufacturing sciences, really, of how to properly isolate T cells, how to introduce the genetic material, how to expand them properly, purify them, and perform quality control prior to their infusion. And the lower left bullet here, referring to regulatory compliance, means, of course, that you can do lots of things in the lab, but if you bring this to the clinic, you must comply with a large set of rules put forth by the Food and Drug Administration in the US. I won't dwell on the history. I'll just say I trace this back certainly for when I went to MIT, it was with the intention of figuring out how to introduce genes into primary T cells, which way back then, perhaps before many of the students listening today were born, this had not been done. Gene transfer biology was just applied to a few mammalian cells. It took a while to figure out how to do it in T cells, but today there are so many different methods that achieve this very effectively. You had to create these CARs, and here I just want to refer to the original in panel A, the original cloning of the CD3 zeta chain, which these three groups independently reported, all creating these fusion molecules that you see here. And that's because the zeta chain, as you know, doesn't have much of an extracellular domain. So they created these fusions so that they could transfect these fusion proteins into T cell leukemias and cross-link them with antibodies to the extracellular domain, and in doing so, discovered that the zeta chain could initiate a calcium influx and T cell activation. Two groups, and most notably the one of Zelig Eshar, then in 2003, replaced these extracellular domains with an scFv, which comprises the heavy and light chains of an immunoglobulin. And they actually call this a T-body, but because we had the means to introduce these in real T cells, primary T cells, we realized that these zeta chain fusions weren't capable of directing a real T cell response, which led us to explore different fusion molecules in T cells, starting with costimulatory receptor. These CCRs and C are chimeric costimulatory receptors. They don't trigger lysis of cells because they don't have an activation domain. I'll come back to those later, which is why I'm mentioning it here. And finally, in D, is what we then named a CAR. which is what I already introduced, and enables for clonal expansion of T cells that engage those cell surface antigens. One needed to identify a molecule, and in our case, we identified CD19, and in 2003, we showed that we could take human peripheral blood T cells, introduce one of these CARs specific to CD19 using a recombinant retrovirus, and upon a single infusion of these T cells, induce long-term remissions in mice. And then finally, you wouldn't go to a company in those days, and there was no industry that was actually interested in this. So you had to do it yourself. And it's my colleague Isabelle Rivière who did that at Sloan Kettering, and two other centers that followed our work and also started CD19 CAR T cell trials already had those facilities in existence at the National Cancer Institute and at the University of Pennsylvania. The first results, turns out, were quite spectacular, very unusual for phase 1 trials in oncology. And on the left here is an example from our first report, which was in adult acute lymphoblastic leukemia, where you see the complete obliteration of these relapsed and refractory ALLs in the marrow of these patients, again, following a single infusion of these T cells. And these results, as you know, were corroborated at other centers. The NCI was the first to report on a patient with a non-Hodgkin lymphoma. University of Pennsylvania was the first to report on 3 patients with chronic lymphocytic leukemia. And we were the first in acute lymphoblastic leukemia. So today, there are 7 of these CAR T cells that are approved in the United States. 5 of them, as you see here, target CD19 using one of these 2 column foundational CAR structures using either the CD28 or 4-1BB signaling domain together with the zeta chain. And 2 of them target BCMA or B cell maturation antigen, which is of particular relevance to multiple myeloma. Now, what I want to discuss with you for the rest of the time are just some more specific aspects of CAR T cells. And I'd like to discuss a little bit the issue of sensitivity of CARs. By sensitivity, I mean how many molecules of the intended target need to be on the surface of a cell, of a tumor cell, for the CAR T cell to effectively engage that cell and go on to kill that cell. So first, this aspect of antigen sensitivity. And it's really from the clinic that this question arose. And here I cite a few papers, actually not from my lab, not from my center, but that reported on this variability in the level of expression of these targets, CD19, CD22, or BCMA. So in the upper left here, you see from Stanford, the study showing you that, for example, the level of CD19 expression in a lot of diffuse large B-cell lymphomas tends to be lower than what you see in a normal B-cell. In lower left, it was noted in our patients and in other patients, including these from this particular report, that some patients respond but then relapse after CD19 CAR therapy. But you will find that sometimes they're CD19 negative, and of course the CAR T cell can no longer perform its task. But more often, they're actually CD19 low. Again, alerting you then to why is it that these T cells could not eliminate these low CD19 molecules. The upper right is even a more clear example in the case of CD22, where you see here the number of molecules, the density of CD22 on a sample of tumor cells from these patients pre-treatment, and at the time of relapse, you see that the density of CD22 is lower. And lastly, this example lower right, in a multiple myeloma patient, you see BCMA expression pre-treatment, complete remission, tumor undetectable, and then finally on the right, relapse, but with lower CD19, lower BCMA at the surface. These observations in patients and these reports drew our attention to the sensitivity of these receptors. And here I bring back the physiological receptor that you see here, the T cell receptor, which you know comprises alpha and beta chains that engage HLA peptide complexes, and it is embedded in the CD3 complex. And we know from many years of literature that this receptor is highly sensitive. It doesn't need that many HLA peptide complexes at the surface of a cell to initiate T cell activation. On the far right here is a CAR, the type of molecules I've been talking about here with the heavy and light chains in yellow and blue. So we wanted to know if we embedded the recognition of antigen of the CAR into a T cell receptor, whether we would get that greater sensitivity into these recombinant molecules. So we created this fusion receptor called HIT, which stands for HIT, but really for HLA-independent T cell receptors. So we reconstruct this by targeting the T cell receptor alpha chain locus, the TRAC locus, in human peripheral blood T cells. And these receptors, as you see, have the heavy and light chains substituting for the alpha and beta chains of a T cell receptor. And we then create panels of tumor cells with different levels of antigen. And so you see here in D, this NALM6 cell line has 27,000 molecules of CD19 on its cell surface, and create these genetic variants that have stable and known numbers of CD19 at their surface, 2,200, or extrapolated to be 20 or so. And you could see here that when we engineered this HIT receptor into T cells, these T cells could recognize tumor cells in vitro in these cytotoxicity assays with very low density, which the best CAR that we could design was unable to do. CARs typically, the CARs that we know today, typically need at least 1,000 molecules on their cell surface, and more often, or often more than 1,000 molecules. I won't dwell on this because this is published. I'll show you a story that is not yet published, where we target CD70 using one of these CARs. And the cancer here is a renal cell carcinoma. It's a patient-derived xenograft. And we introduce it either in the lung of these mice, these NSG immunocompetent mice, or in the tumor itself. And in fact, when the tumor is in the lung, our CAR T cell that's CD70 specific is very effective with 1 million CAR T cells. But when the same PDX model is in the kidney, the CAR fails. And in fact, we can even go to these very extravagant, I consider extravagant doses of 5 and 10 million CAR T cells in a mouse, and it still fails. At first, we thought it was a failure of the CAR T cells to enter the kidney, but that was ruled out. And we soon realized it has to do with the antigen density. Here, you see an immunohistochemistry of this PDX in the kidney, and you see brown cells that are positive and a lot of blue cells there are that dots that appear to be negative. When you take out these tumors from these mice, using one of these two PDX models, K5 or K7, you see that the CD70 expression is bright in the lung, but it's clearly low and negative in the kidney for this PDX or also this other PDX. So CD70 appears to be absent at the kidney site, although not at the lung site. And we thought at first that maybe these tumors were negative for CD70, but it turns out in the kidney, but it turns out they're not negative. They express CD70 at a very low level, and tested the HIT receptor. And if I turn to the BLI here, you can see that, again, those purple and blue lines here are the failure of the best conventional CAR that we could design in treating this PDX at the kidney site. But you see, when we infuse the HIT T cell, there's a rapid decrease in tumor burden. They do relapse because these T cells don't have a very long lifespan. But if we endow the HIT T cell with forms of co-stimulatory support, one way we have of doing this in laboratory experiments is by co-expressing CD80 and 4-1BB ligand. We now see that with the HIT receptor, we achieve this durable and complete remission. Now, another aspect of sensitivity that I'll only briefly mention is, of course, you might have thought, mediated by the affinity of the scFv that is incorporated into the CAR. And so this work is mentioned, and I'll just briefly highlight it. We were curious to know how these CD19 CARs, of course, bind to CD19. So finally, we teamed up with Joel Meyerson, who was at Cornell University at the time. And through the cryo-EM that he and Cheng Hao performed, we saw where these two scFvs bind to CD19. The SJ25C1 is the one that I consider the original CD19 CAR that we reported on, but most others who followed used the FMC63 scFv. You can see that they actually bind to an overlapping site on CD19. But really, with this information and using some modeling, ascribing different forces to different amino acid, we created a series of mutants, again, informed by this structure. And all I want to show you here is that with that information, you can rapidly calibrate not just the affinity of the CAR, but the sensitivity of your CAR T cell. So again, this panel, I showed you NALM6 with 20,000 molecules per cell, another one with 2,000, another one with 200. These are in vitro assays, and we introduced the CARs actually to the track locus. So there's all same copy number and promoter in these human primary T cells. I'll just take almost a random example here. It's one of the mutants in the orange. You can see that this CAR T cell nicely kills the 20,000 CAR T cell. It's a little less effective on the 2,000, clearly. And as you see, as the density drops to 200, in vitro, it's ineffective. Needless to say, it completely fails in vivo. So you can also control the sensitivity of your CAR by adjusting the affinity of your scFv. Next, I want to talk about logic gating of T cells. And this language has now been adopted in the field to describe different ways that you may pursue to target more than one antigen. Again, the T cell receptor that I mentioned at the very beginning is clonotypic. There's one T cell receptor per T cell. But in this world of synthetic immunology, you don't need to restrict yourself to one. You might create T cells that engage two antigens, maybe 3, 4, or more. But just keeping it to 2 antigens, there are many ways of doing that. And this Boolean nomenclature fits rather well. One is an OR gate. So imagine a T cell that can target antigen A or antigen B. This is the most commonly explored gate. Obviously, it expands the reach of the CAR T cell to 2 antigens and not only one. Another engineering approach, at least in principle, is what you would call an AND gate. This would be a T cell that would only activate in the presence of both A and B on the same T cell, but not if A or B were found alone. We published over a decade ago a first version of this. It's not easy to design. Robbie Meisner last year published an improved version of this. It's a difficult gate design, but it would be very interesting if it worked well, because if you lack tumor-specific antigens, like A here is found on some normal cells and B is found on some other normal cells, you could create some tumor specificity for A and B together in some settings. A NOT gate is the term used today for what we called many years ago inhibitory CARs. We had reported on CARs based on PD-1, which here would recognize, say, antigen B, that in principle could protect cells that express B, but allow for the CAR that's anti-A to proceed on a tumor cell that does not have B. Very interesting gate as well, not easy to design. In D is another gate called if-then. The best known version of this is Wendell Lim's SYN-Notch receptor system. So the receptor engaging A here is not a CAR, but upon binding to CAR, it induces transcription. Binding to antigen A, it induces the transcription of a CAR specific to antigen B. So this is a temporal gate. It takes time to induce the CAR at the cell surface. And the idea here is to not constitutively express the CAR anti-B for safety reasons, and maybe also for avoiding what's called tonic signaling of constitutively expressed CAR. And the fifth gate here is what we call an if better gate. That's the one I want to show in a few minutes. This is a helper gate. So imagine here a CAR that engages antigen A, but it doesn't do well when the density of A is very low. And maybe that's fine if there's low expression of A on normal cells, but then the tumor would escape. So can you engage a second molecule that is found on tumors but not normal cells that would serve as a helper molecule to the CAR, which here engages antigen A? And I will show you how we have been developing this. And here I turn to a project focusing on acute myeloid leukemia, where we at first surveyed expression of different candidate target molecules in primary samples of patients with relapsed refractory AML. And Sasha monitored in particular expression of CLEC12A, AGRA2, CD123, and here CD33. And looking at this, and the y-axis here are not the mean fluorescence intensity, but the median number of those candidate targets measured in these patient samples. You may be drawn to target CD123 and CD33 owing to their higher expression, and indeed there are today many trials targeting those two antigens. But we elected not to pursue this for the reasons shown here on the right. And this is normal bone marrow. And you can see that there is also higher expression of CD123 and CD33 in a number of normal cells, which include some active hematopoietic stem cells and common myeloid progenitors in particular. And we didn't think that we could target these high cells while sparing these that have 1,000 or more of these molecules on their surface. We thought we would have a better chance with CLEC12A and AGR2. You see here in the normal, there is AGR2 expression on common myeloid progenitors, rarely exceeding 1,000 molecules per cell. And there is CLEC12 expression on, neutrophils and monocytes. So we thought we could test a CAR targeting AGR2 that wouldn't be too sensitive, because we want to spare these cells here that I'm highlighting in green, but use then this chimeric co-stimulatory receptor that I mentioned at the very beginning, and that you see here does not have a zeta chain, does not initiate cytotoxicity against CLEC12A expressing cells, but can bind to CLEC12A when it's there. So how does this work? I'll just highlight some of the key in vivo studies. This work was published here last year. And so MolM13 is shown here. It's a much used AML cell line. You see that orange dot here, so you can see where it fits on this spectrum of patient samples. Now, MolM13 has about 10,000 molecules of AGR2 on its surface. And so we infuse these NSG mice harboring MolM13 with actually not a very high dose of these CAR T cells. The CAR T cells express the CAR against antigen A, or here AGR2, and they express the CCR, the chimeric co-stimulatory receptor, against antigen B, which here is CLEC12A. And actually, these CAR T cells are very effective. You might say, well, did CLEC12A make a difference? Presumably not, but we checked that. If you eliminate it and create now this variant, now you see this orange dot has moved down here. The CAR T cells still work very well. And indeed, 10,000 molecules of AGR2 is a good number of targets. But now let's create another variant cell line. Now we have 1,000 molecules of AGR2. And you see the orange dot now where it is. It's at this position here. And now these CAR T cells fail, which is exactly what we wanted to see. And now we can ask the if better question. So now we have a variant of this line that has 1,000 molecules of AGR2, but a higher number of antigen B. And now the CAR T cells work again, if I can say. This is the if better gate. This is failure to recognize 1,000 molecules of AGR2 alone, but successfully eliminating the tumor in the presence of the second antigen. So we're now running a clinical trial at MSK, or at least my colleagues there are doing that now. And we will see if Bettergate works in patients as well as it does in animal models. Next, I would like to turn to some aspects of T-cell persistence and address the CAR signaling, and after some aspects of allogeneic CAR T-cells. The clinic, once again, gave us some clues as to, the impact of strength of signaling. And so the field quickly observed that CD28 CAR T cells are highly effective, but short-lived. In contrast to the 4-1BB CAR T cells, which are less potent as effectors, but on the other hand, persist longer. And at first we had thought that, well, those were just attributes of CD28 and 4-1BB. But then we asked ourselves if, in fact, this molecule shown here, which is the CD28 CARs that are used all over the world now, and that we had reported long ago, were wanting to increase strength of signaling, were in fact providing too strong a signal. And so we revisited them by focusing on the zeta chain and these activation motifs, the 3 ITAMs that are well known to be in the zeta chain. And we know from the mouse literature how to introduce point mutations that inactivate these ITAMs one by one. Anyway, this was published years ago, and in this paper we showed, and here's just an in vivo data, showing you that in a model of acute lymphoblastic leukemia that's CD19 positive, the Nalm-6 cell line is born by these mice. A single infusion of the low dose of CAR T cells, purposefully chosen to be a low dose of this CAR that is, again, has been administered now to tens of thousands of patients, but here used purposefully at a low dose, doesn't really cure these mice. And here are 3 CARs with 1 ITAM only, and it's the one with the proximal ITAM, which we call now 1XX, that provided the best responses. And in this paper, we provided a model explanation for this, saying that this 1 ITAM combined with the CD28 cytoplasmic domain provided strength of activation sufficient to confer good effector functions to these T cells, but not so strong as to turn off their memory or persistence, if you like, programs. What I really want to discuss is what we've done since, which was to bring this to a clinical trial to see if really this 1XX trial is better than the 2 CAR T cell trials that are used, the 2 CAR designs that are used so often that I showed you in my very first slide. So we're targeting here CD19 using this scFv that binds to CD19 shown up here in patients with relapsed refractory diffuse large B-cell lymphoma. What I want to address here is the T cell dose. You see the doses that are used with the 2 more comparable, bulk T cell products that are FDA approved. This T-Sagen here is a 4-1BB CAR, typically requires 300 million CAR T cells in adults. The C28 CAR requires a bit less, 120 to 160 million CAR T cells in these patients. Now, we thought, well, if the 1XX CAR is a better CAR insofar that has good effector functions but persists longer, it should work at a lower T cell dose and provide at least equivalent outcomes in these patients, but with a lower T cell dose. So here's the dose escalation scheme starting from 25 million per patient and up. And here's on the left bar graph showing you the complete response rates in the 4 dose levels. You see that are pretty good, actually. If you remember, DLBCL is the harder to treat of all the B-cell malignancies. Small numbers, of course, it's a phase 1 trial, but most notably, no dose response, really. And so we thought that we would not do the conventional thing of expanding cohorts at the maximum tolerated dose, but rather at the lower one, since that seems to be sufficient, 25 million. And you see in the altogether 16 patients that we treated at the level of 25 million CAR T cells, the complete response rate is very good of 75%. But also our second prediction is that treating patients with lower T cell doses would result in lower toxicities. And that's indeed the case. Not a single report of a meaningful neurotoxicity and only one instance of what's called a cytokine release syndrome. We waited a long time to publish this work, which we just published, I think, last month, because we wanted to know the durability of these responses, and they are durable. Most of these patients are 2 years out. So truly, 25 million of a 1XX CAR T cell is sufficient for these diseases, in contrast with the doses that I showed you before for the earlier versions of CARs. And just quickly, I don't have time to dwell on this, but I want to refer to the work of two other groups who were interested in testing our 1XX CAR. Jonas Madsen, who tested it in mesothelin-targeted CARs in mice and models of orthotopic ovarian cancer, and found that the 1XX CAR did outperform the 28 and 41BB CARs in those models. And also the work of Cynthia Bamdad and her colleagues in breast cancer, targeting another antigen called MUC1 star, where they too again found that the 1XX CAR T cell, here shown in green, could outperform the two more conventional CAR designs in this cancer model as well. Next, I want to address another aspect of persistence, but in a very different way, in the context of allogeneic cell therapies. And here, I just want to say a word about the T cell sources used for these therapies. Obviously, these therapies were at first autologous, and the overwhelming majority of these trials are still autologous. Now, that's ex vivo manufacturing. Some have said that this might be phased out. I'm not so sure about that. I think these methods work well, they're robust, and importantly, they're becoming shorter and shorter. The first cultures required, 2 weeks of culture to produce these cells. All of our protocols in the last years are a 7-day manufacturing process, and that at many other centers as well. Some have now shortened it to 3 days, and some report an overnight process. And then furthermore, I just showed you the dose reduction that you could achieve with 1XX. You don't need 300 million CAR T cells, you need 25 million. That's 10 times less. And today, some industries are trying to robotize and miniaturize this. So I'm not convinced the least that ex vivo would be phased out. I'll come back to in vivo in a minute, very briefly, but I'll come back to it, which is obviously autologous, and just turn here to allogeneic cells, which obviously would be a great solution to have cells that from one donor or one source that you could administer to multiple recipients, and that could be prepared in advance, off the shelf, as one says. Two main sources are either cells harvested from healthy donors or cells that are produced from pluripotent stem cells, T cells that never developed in a thymus or in the body of a human being, but are derived from pluripotent stem cells. And there are several groups making good progress towards generating, effective T cells in vitro. But what I want to discuss briefly today here is just the challenge of whatever your source of allogeneic T cells, they will face immune rejection, either antibody-mediated or NK cell-mediated, especially if you ablate expression of HLA class 1, and also CD8-mediated T cell rejection by the host, obviously rejecting the medicine that they are receiving, the allogeneic CAR T cell. So the question is, how can we reduce this allogeneic rejection of incoming CAR T cells by the host, especially without causing massive immunosuppression of the host, which might help the CAR T cell, but leads to very bad complications, infectious complications in those patients. So how could we just at least extend the survival of allogeneic CAR T cells? in recipients. And so I want to highlight here the work of Carlo Perica. And first of all, and first and foremost, perhaps the model that he established. And here's several groups have investigated possible allogeneic approaches, but we felt it needed a more robust model. And here's the one we came up with. So these are, again, immunodeficient mice. The readout is the efficacy of CD19 targeted CAR T cells. But prior to infusing these CAR T cells, the day before, we replete these mice with either autologous or fully mismatched allogeneic PMCs, peripheral blood mononuclear cells. And they are completely mismatched. And the key detail here is the dose. There's 5 million CAR T cells to every mouse. 5 million PBMCs administered to every mouse. And then 10 to the 5 CAR T cells. So it's a 50-to-1 ratio. Now, if you do that in a mouse like that, and you measure the CAR T cells on day 10, which I show you here, these in the presence of these allogeneic PBMCs, they've become already hard to detect. If you deplete the CD8 T cells from PBMCs, you see the numbers are restored. The impact of depleting either NK cells or CD4 cells is modest. So this work is in press. The paper is online, and the final paper should be coming out very soon. It describes how we started not ablating HLA class 1, but reducing it by exploring the immunoevasins of a number of viruses, CMV, EBV, Kaposi sarcoma virus, and HIV. And in doing so, we found that HIV-NEF had the most interesting properties. And here I highlight an example of that. And that is in these NSG mice. In this case, they also have IL-15 because we want to sustain the NK cell activity of the PBMC, that's autologous or allogeneic. And you see here the tumor progression. And so again, the readout is the activity of these allogeneic CAR T cells. And in red here is the what happens if you knock out class 1 in the incoming CAR T cells. And you can see that these T cells are very rapidly rejected. They perform even less well than the controls, straight up allogeneic cell, which are all these mice in black. You see the survival below. But if we co-express NEF in these cells, which we show reduces class 1 expression, but doesn't abolish it, the activity of these CAR T cells is much more sustained. The reduction of class 1 expression is to about 15–20% or so of baseline, which is enough to slow down the CD8-mediated T cell rejection of those CAR T cells, but not enough to render these cells sensitive to NK cells. And here's another series of experiments where we're co-expressing NEF in 1 CAR T cells. And I just want to show you in this, again, in the leukemia model targeting CD19, that by 2 weeks, there's actually 50 to 100-fold more of these allogeneic NEF expressing 1XX CAR T cells than the control that doesn't express, which is substantially more and accounts for the better response in these mice. Now note that ultimately these cells will be rejected. And so what we're really doing here is buying time. Lastly, I want to touch on in vivo T cell engineering, and I'll be very brief. We've only recently started working on this, and others have been working on this for some years. But it has become today a new and other exciting direction in this field. There are 2 main approaches to go in vivo. Some you make use of engineered lentiviral vectors, and others of lipid nanoparticles. And this work is already today reaching the stage of the first clinical trials. The next generation of this really, though, is to target these devices, obviously to try to target them to T cells or other immune effector cells. And here I just want to refer to the work of 2. There's many working this field, but these 2 worked in my lab before. And Justin Equem recently evolved the capsid of AAV vectors so as to specifically target mouse T cells and created in this way an amazing tool to start engineering primary mouse T cells. First ex vivo, but I know that he's working on it in vivo. And Matthias Stephan, years ago, after leaving the lab, started working on lipid nanoparticles, and he targeted them with an scFv, targeting them to CD3. And I think there's much more to learn about these targeted gene deliveries. I refer to another one here that we are working on now, where a lentiviral vector is coated with polymers to which different targeting agents can be attached. So an exciting new direction for the field, and let's see what comes out of it. So of course, all of this leads ultimately to new applications. And here you see the tip of the iceberg, CD19 CAR therapy in lymphoma and leukemia. Today, myeloma has also some approved CAR therapies. There's still room for progress. We know that solid tumors have not been yet cracked, but I'm hoping, of course, that as we better understand how to engineer a functional, durable T cell and find the right targets and gate them in the right way, that we will get there. And of course, other directions. And autoimmunity is the one that I want to finish on. The group of George Schett presented exciting initial results in patients with lupus, where the side effect of CD19 CAR T cells in cancer patients, which is the B-cell aplasia, the side effect in cancer now is the therapeutic effect in patients with autoimmunity. And this is a table that we don't have time to go through, but it makes just one point. There are a lot of trials today in patients with autoimmunity using CD19 CAR T cells exactly how they were created for lymphoma and leukemia, using different binders and different signaling domains. And I think there's a whole field now that has opened in lupus, but also a number of other autoimmune disorders. And with that said, I will stop here. And I thank you for your attention.